Hello and welcome to biology class. We are studying topic biochemistry of cell, and in this topic we are dealing with biomolecules which are present inside the cell. So how many biomolecules are there inside our cell? 4 biomolecules. Which are those? Carbohydrates, then proteins, third one is lipids, and fourth one is nucleic acids. So first 3 of them we have already studied. We have studied about carbohydrates, we have studied about proteins, and we have even studied about lipids. We have studied about chemical structure of these molecules as well as type and classification of these molecules. So today we are going to shift to the last molecule of our studies, that is nucleic acids. Now nucleic acid, they are the last one, but they are very important one. So let us begin studying nucleic acids in details. Now, when we talk about nucleic acid, when I say nucleic acid, nowadays this is the most studied and most interesting biomolecule. When I'll teach you in details, you will also develop interest in studying nucleic acid. Now, when we consider nucleic acids, the nucleic acid term or nucleic acid was first introduced to us or introduced to human being by this person. This is a scientist called as Friedrich Michel. Now this scientist is from a country of Switzerland. At the age of 21, when he was 21 years old, he discovered nucleic acid for the first time, and that was the year 1869. So in 1869, he told us about nucleic acid. Now what exactly he told and what exactly he studied? Let me tell you in short. Actually when he was a college student, that time he was studying human cells. Okay, and that time see the year 1869. It was long back. That time we were not knowing much about nucleus, not knowing much about cell in details. So that time he told nucleus of the cell contains some acidic compound. What he said? Nucleus contains some acidic compounds. Acidic compounds. And those compounds are acidic and present inside the nucleus. That's why he named it as a nuclein. What does he name? Nuclein. Now these nuclein afterward named as a nucleic acids. Okay, and he even told that this nuclein must be responsible for carrying information from parents to offspring, which we called as a hereditary information. So this nuclein is also responsible for heredity, and all these things today we are very clear about it, and we know everything that now we know DNA is our hereditary material and DNA is an example of nucleic acid. So in this topic basically we are going to study nucleic acid in details as well as DNA and RNA in details. So let us begin our studies properly. Now when we consider this nucleic acid, this diagram show nucleic acid. Now when we consider nucleic acid, they are the very important biomolecule, and that's why they are located in extremely important organelle inside our cell, that is nucleus. Now this is a diagram of a cell culture of a human cell culture, which is called as HeLa cells. We are not dealing with HeLa cells in detail. HeLa cell is having its own story that I will tell you some other day. Now when we consider this particular picture, now here the nucleus and particularly DNA in the nucleus is stained with blue color. So you can see this grey colored cell, and inside this a blue colored nucleus, and in that more the blue color, more is the DNA. So we can see lot of DNA, that is lot of nucleic acid inside the nucleus. Now located inside the nucleus more, but even they are present in the cytoplasm. Now see, when we talk about nucleic acid, when we see nucleic acids, There are two types of nucleic acids which we are going to study in this topic. One is called as DNA. One type of nucleic acid is called as DNA and another type of nucleic acid is called as RNA. Both the nucleic acid we are going to study in details. Now let us start studying their full form first. When I consider DNA, full form of the DNA is deoxyribonucleic acid. Nucleic acid. Actually, deoxyribonucleic is one word. I wrote it separately just to understand the full form. DNA. Deoxyribonucleic acid. DNA is present inside the nucleus of eukaryotic cell. DNA is present in the nucleus of eukaryotic cell. Clear about it? Now, another thing is that when I talk about RNA, another type of nucleic acid, the full form of RNA is ribose nucleic acid. Ribose nucleic acid. And basically, this ribose nucleic acid found in both nucleus, it is also found in nucleus as well as it is also found in cytoplasm. Understanding? So, nucleic acid basically two types, DNA and RNA. DNA, deoxyribonucleic acid, found in the nucleus of the eukaryotic cell. Even it is present in prokaryotic cell, but it is not not enclosed in the nucleus. It is present just in the cytoplasm. Then when we consider RNA, full form is ribose nucleic acid and it is present in nucleus as well as in cytoplasm. So let us go in more details about the nucleic acids. First we will try to understand the chemical structure and then we will move to its function and types. Okay, as usual. Now when we consider nucleic acids, when we talk about nucleic acids are also polymers. What they are? Polymers. Just like other biomolecule except lipids. Correct? Now you tell me what is polymer? Polymer is the compounds or biomolecules which are made up of small units, small monomers, correct? So polymers are made up of monomers. Likewise, we have studied carbohydrate. Basically polysaccharides are made up of small glucose units. Proteins are made up of many amino acids joining together. Likewise, nucleic acids are also polymers and they are also made up of small units joined together. The name of the monomer of nucleic acid is nothing but nucleotide. Nucleotide. So, nucleic acids, that is DNA and RNA, both are formed by joining these nucleotides together. Thousands of nucleotides join together to form nucleic acid. Understanding? Now, we will study first while studying nucleic acid. First, we will study about nucleotide, the chemical structure of nucleotide, and then we will shift to the how exactly they join, how exactly their bonding and other things. Okay, so let us begin by studying the structure of nucleotide. So I'll just draw a structure of nucleotide. Structure of nucleotide. Now I will draw the structure and I will explain you. Now when we draw nucleotide structure, we draw it something like this. This should be exact circle. Excuse me for my drawing. See, this is how usually nucleotide is constructed, or nucleotide is drawn, or it is represented. Now try to understand what this each component is. Now here you can see this first structure, that is this circular structure, which is nothing but a phosphate group. I am just indicating it as a P. Then here the second component from this is how the second component. Okay. Now second component is called as the sugar component. Okay. And this third component is called as the nitrogenous base. See, three component. First one phosphoric acid, second one a sugar component, and third one nitrogenous base. We will study each component in detail. First we will enlist the component and then we will study each component in details. But have you got the structure? Can you draw it now? Remember, you need to draw it again and again while studying this topic, as well as it will come several times in your 12th standard. So just practice drawing it. One circle, it is joined with this rectangular structure to the sugar, and then sugar is joined to the nitrogenous base. We will draw it again and again. By practice you can easily draw it. Don't worry about it. But right now just remember. Okay. Now let us go ahead. Now let us study each component in detail. So first component of the nucleotide. First component of nucleotide. Remember, nucleotide is a monomer of nucleic acid. So many nucleotides join together to form nucleic acid, and we are studying nucleotide in detail. Whatever structure I have drawn, that structure is nothing but a structure of nucleotide, a single nucleotide, one nucleotide. Got it? Now let me enlist the components and we'll go ahead. The first component is phosphate group. Okay. The second component is pentose sugar. Pentose sugar. We will talk in details about pentose sugar after some time. Then third component is nitrogenous base. This nitrogenous base. Got it? Clear about it? Let's go and study each one of them in detail. Now the first component, let us start with the first component. First component is phosphate group. Phosphate group. Now when I consider this phosphate group, actually it is a phosphoric acid. But when it come and join to the nucleotide, that time just phosphate group is attached. So phosphate group is something in which at the center there is a present phosphorus, and to each side of the phosphorus they are attached oxygen. One oxygen is attached with the double bond and another one is attached with, oxygen is there but as a OH group. Understanding? So phosphate group is something like center phosphorus, and to all the sides of phosphorus there is the attachment of oxygen, and one side OH group. Like oxygen is there but its hydrogen is also there, and each of these oxygen is having negative charge. Oxygen is having negative charge. Okay, got it clear? So this is about phosphate group, and we just draw it like a circle, one circle. This much is enough for you, and just you need to remember, to every side of phosphorus there is the attachment of oxygen. Okay. So let us go to the next component, which you need to understand in more details to properly understand DNA and RNA. See, the second component of nucleotide is pentose sugar. Pentose sugar. Now, when we have already studied carbohydrates, right? In the carbohydrate, we have already studied pentose sugar. Do you remember it? What is pentose sugar? A sugar with 5 carbon, correct? The sugar with 5 carbon is called as the pentose sugar. We have studied in simple carbohydrates, correct? So now there are several different type of pentose sugar. The one which is present in the DNA, the one which is present in the nucleic acid in general or nucleotide in general is called as ribose sugar. What does it call? Ribose sugar. Okay. Now, let us try to understand the structure of ribose sugar first, and then we will study the details of it. Now, when we draw the ribose sugar in the nucleotide, we have drawn something like this. See, it was having oxygen at one side, correct? This is something we have drawn, correct, as a sugar. Then it was having phosphate on one side, correct? But this is a pentose sugar. Now, let me draw a chemical structure in detail so that you will understand it properly. So, how to draw ribose sugar, that you try to understand. Now, each of the corner indicate carbon. Okay. Now, you need to draw oxygen first, then attach it with the carbon. This corner carbon. See, here one carbon, here another one, and one more here. Okay. Now, so carbon, then here one more carbon, one more, and these two are attached, and fifth carbon is over here. Okay, got it? Now, each of the carbon is having other groups also. This carbon will be having OH and H group. This carbon is also having OH and H group. This carbon is also having OH and H group. And this carbon at one side, it is having another carbon to which CH2OH is attached. Okay, and to this carbon one more H is attached. Now, this is a chemical structure of ribose. Try to understand it properly. You are having this in details. See, how to draw ribose? Draw oxygen to which attach 5 carbons. Okay, this is the one more. Okay, it is a pentagonal structure. Okay, and to every carbon there is attachment of C and OH group, and to this carbon, the fourth one, it is attached as a CH2OH group. Okay. Now, let me number the carbon so you will understand it properly. From the oxygen, we need to move like a clockwise direction. So, this will be the first carbon, this will be the second carbon, this is the third one, this one is the fourth, and this one is the fifth carbon. Okay, this numbering the carbon is very, very important. See, in the clockwise direction from oxygen we need to start. Carbon 1, carbon 2, carbon 3, carbon 4, and carbon 5. Clear about it? So, this is the structure of ribose. You need to understand it properly. See, let me repeat. Ribose is a 5-carbon sugar, okay, which show this pentagonal structure. To one side of the one angle of the pentagon there is oxygen, okay, and other 4 there are carbon, and one more carbon is attached in the straight line above from the pentagon, okay, and to which H and OH groups are attached, except for the 4th and 5th carbon. Carbons are numbered in a clockwise direction from oxygen. Okay, got it? So this is the structure of ribose sugar. There is one variation of sugar in the nucleic acid. There is one more type of sugar present in the nucleic acid. See, that is also similar to that of the ribose sugar. Just there is one difference. Let me draw the structure and let me tell you the difference. See how I am drawing it. Carefully watch it. H2OH. Here there will be H. First carbon H, OH. Second carbon there will be H. Third carbon there will be H. Okay. And now see the carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. Clear about it? To the first carbon, H and OH group, just like a ribose sugar. To the second carbon, but there is a H group just like ribose sugar, but to the second carbon, instead of OH, instead of this OH, here in this sugar there is only H. Clear about it? Third carbon is as same as the ribose. Fourth is also as same as the ribose. Fifth one is also same. Only difference is in the second carbon, where OH of the second carbon is replaced by H. Clear? So what happened? So this is also ribose sugar, just little bit different ribose sugar. In the second, see, this is the first diagram and this is the second ribose sugar. So in the second one oxygen is removed. So it becomes deoxy. What it become? Deoxy. D means what? Removed. And oxy means what? Oxygen. So from the ribose sugar, one oxygen is removed. So it became deoxyribose sugar. Okay. Now pay attention properly. So we are having two types of sugars, two types of 5-carbon sugars present in the nucleotide. One is the ribose sugar. This ribose sugar is present in RNA. Ribose nucleic acid. This ribose came from ribose sugar. And this another type of another type of sugar is present in DNA. DNA full form is what? Deoxyribose nucleic acid. This D came from deoxyribose. Understanding? Clear about it? And why deoxyribose? Because one oxygen from second position is removed. Okay, so it became deoxy. Now this deoxyribose is very stable. It is very stable. That's why DNA is our genetic material. Those kind of things we are going to learn next year. But this year you need to understand the chemical structure. See, nucleotide we are going to study. What is nucleotide? Nucleotide is the monomer of nucleic acid. Each nucleotide structure we are going to study in detail. So nucleotide, when we consider, it looks something like this. We have studied about its first component, that is phosphoric acid. Then second component is sugar, which is pentose sugar. Now we will shift to the third component. So first and second component are clear. Phosphate group is nothing but came from the phosphoric acid. Then sugar is nothing but the ribose sugar. In RNA it is ribose and in DNA it is deoxyribose. Clear about it? So we'll shift to the third component, nitrogenous bases. Okay, let us begin to study nitrogenous base, which is nothing but the third component of nucleotide. So let me take a read of this and we'll go ahead. Third component is nothing but nitrogenous base. Nitrogenous base. Now try to understand this nitrogenous base. It is a little bit interesting. Now base means it is basic in nature. It is a basic compound. Okay, means pH is more than 7. Okay, then it is a nitrogenous means nitrogen containing base. Nitrogen containing base. It is nitrogen containing basic compound. Okay. Now when scientists studied these nitrogenous bases, they found that these nitrogenous bases are ring compound. Means they form a ring like this. They form ring and this ring contain nitrogen. Okay. We are not going to study in details where exactly the nitrogens are present. It will become very, very difficult. So we are going to study simple thing. So remember, nitrogenous bases are nothing but nitrogen containing compounds. Okay. And they form ring structure. Okay. Now two types of rings are observed in the nucleotide. One ring is very simple like this hexagonal ring, and another ring is quite complicated. It is a double ring. How it is? Just try to understand. It is also having one hexagonal ring like this. Okay, and to this hexagonal ring another pentagonal structure is attached. Another pentagonal ring is attached, better to say, like this. Just a second, let me erase this. See, this is hexagonal ring 1, 2, 3, 4, 5 and 6, and this is pentagonal ring 1, 2, 3, 4 and 5. Got it? So, and this is also hexagonal ring. Hexagonal means having 6 corners. Pentagonal means having 5 corners. Okay? It is that simple. Things are very simple in biology basically. Just you need to understand words properly and you need to remember a lot. Okay? So, there are two different nitrogenous bases. There are two different type of nitrogenous bases found into the nucleotide. One are very simple with single ring structure and another are little bit complicated structure with 2 rings. Okay. Now the single ring structure nitrogenous bases are called as pyrimidine. What does they are called as? Pyrimidine. Single ring, pyrimidine. Double ring are called as purines. Single ring, pyrimidine. Double ring, purine. Okay. Now when we consider this pyrimidine, there are 3 different type of pyrimidines found in nucleotide. 3 different types. Okay. First type of pyrimidine is called as the cytosine. It is called as the cytosine. Second type is called as second one is called as the thymine. And third one is called as the uracil. See, there are 3 different pyrimidines present in the nucleotide: cytosine, thymine and uracil. And there are only 2 different type of purines. One is called as the adenine and second one is called as the guanine. See, this is the very extremely important thing you need to remember. Nitrogenous bases, 2 types of nitrogenous bases, pyrimidine and purine. Okay. Pyrimidine, complicated name but simple structure, single ring. Purine, simple name but complicated structure, that is double ring structure. There are 3 different pyrimidines present: cytosine, thymine and uracil. Okay. There are 2 different purines are present: adenine and guanine. Now this is the extremely important thing you need to remember in this topic. Okay. Very, very important to remember. Never ever forget which are the purines and which are the pyrimidines. Now it is really difficult to remember this, which are the purines and which are the pyrimidines. Now there are 2 ways to remember this. One way how I do remember this is that, see, pyrimidine is a complex spelling, simple structure. Complex spelling, simple structure. Purine is a simple spelling, complex structure. Okay, this is how I remember which are the single ring and which are the double ring. Now how to remember the examples, which are the purines and which are the pyrimidines. Okay. Now there is one sentence to remember this. It's like it's the statement CUT, cut pi, means C is for cytosine, U is for uracil, and T is for thymine, are pyrimidine. See, cut pi. How we can elaborate it? Cytosine, uracil and thymine are pyrimidines. Okay. And for remembering name of purines, there is a statement is like angels and gods are pure. Angels and gods are pure. A and G are purines. Okay. And remember this properly. Cut pi and angel and gods are pure. CUT means cytosine, uracil and thymine are pyrimidines, and A and G, that is adenine and guanine, are purines. Okay, basically they are abbreviated as letters, means cytosine always abbreviated as C, thymine as capital T, and uracil as a capital U, capital A as adenine, and capital G as guanine. And it is extremely important to remember them. Now these three components, that is phosphoric acid, pentose sugar, and nitrogenous base, join together to form a nucleotide. How they join together to form a nucleotide, as well as how different nucleotides join to each other, that all the things we will study in the next lecture. Till then, goodbye, take care.